We're going to talk about assembly of virus particles. This is the second to the last lecture on the reproductive cycle. On Monday we'll talk about metabolic changes in cells, and today we're going to kind of culminate all of the things we've talked about and build virus particles using nucleic acids that we've already reproduced in the proteins that have been made. And we've talked about a lot of different virus particles so far in terms of their morphology. You know, we have naked icosahedral particles or enveloped icosahedral particles. We have envelope particles with helical nucleocapsids. And we'll talk about the different ways that these different kinds of viruses particles are put together today. All virus particles have a common set of reactions that they undergo in order to be built, and they're listed here. You start with formation of individual units of the protein shell. We talked about structure and how you have polypeptide subunits which assemble into larger and larger structures. Assembly of the shell by interactions among the subunits, which we talked about before. Packaging of the nucleic acid. We haven't talked about this very much, and we're going to discuss it today. Selective packaging is a key here because you don't want to package cellular nucleic acids. As I said before, that would be a waste. It would not be infectious. Now, some viruses acquire an envelope, a process that we will talk about today. Viruses are then released from the host cell. And, you know, we mostly talk about mammalian viruses, but there are viruses of yeast which are never released from the cell. They're simply passed from yeast to yeast as they divide. So that's obviously not a requirement for all viruses, but most of the ones we talk about in this course. And sometimes after virus particles are released, they mature. We're learning more and more examples of that. We always thought they mature inside the cell, but now we know examples of maturing after the release, and we'll talk about one today. Now, assembly does not happen without host cell machinery. Even though we could make a capsid protein and it might assemble in vitro, all the other steps do not occur without a cell. The cell provides many things. Of course, these are obligate intracellular parasites. So the cell provides many things, including for assembly chaperones, that is, proteins that assist in assembly reactions. Transport systems, not only to bring the viruses into the cell, as we talked about during entry, but to bring them out or on the way out. Secretory pathway is very important for envelope viruses, as we'll see, and nuclear import and export machinery. Many viruses are assembled in the nucleus, not all of them, but many of them are, and they have to get components in and then back out again. So the import and export machinery, which governs how things move in and out of the nuclear pores, is very important. These are just a few of the things that are needed for assembly. And as I said earlier, in an early lecture, things don't diffuse in virus-infected cells. Components do not simply diffuse, they move by directed transport pathways. And these can include pathways involving short distances, and these are typically channels, protein channels that move things through them from one side to another. So here's a protein channel in a membrane, and viruses or viral components can move in and out of those short distances, angstroms to nanometers. Then we have long distance movement. Of course, when a virus particle has to go from the plasma membrane to the nucleus, that's a long way, and you have to go on cytoskeletal tracks with motors that require energy. And there we're talking about micrometers to meters, and for example, moving, as I said, from the plasma membrane to other sites in the cell. And this immunofluorescent image on the right is an example of that. This is a cell infected with vesicular stomatitis virus. And the cell is stained with a few different stains. One is DAPI, which stains DNA, so that's blue. You can see the nucleus. And then there's an antibody to tubulin, which stains the microtubule network, the mesh on which virus components move, endosomes and virus components. And then the yellow is a stain for the nucleocapsid protein, which needs to move around the cell in order for virus particles to be built. And you can see this nucleocapsid protein is associated with the microtubule network. It moves through the cell on the microtubules. Now the bottom cell has been treated with a compound called nacatazole, which disrupts the microtubules. And you can see in red, there's no longer the nice filamentous microtubule network. And the viral end protein, nucleocapsid protein, is stuck at very specific places. It can't move around because the microtubule network's been destroyed. So they're stuck in little patches, which clearly are very different from the untreated cell. And that cell will not produce infectious virus because the nucleocapsid can't move around. So a simple example of how movement in cells is very important for viral reproduction. A key take-home message, which I like to say, is that nothing happens fast in dilute solutions. And during virus replication, components are often concentrated so that reactions proceed more quickly. And because of this, we can often see things happening in infected cells by a light microscope. We can't see virus particles very much, except for the biggest viruses, but we can see components. We call these factories or inclusions. And so many viruses build particles in very focal sites because they can concentrate all the materials that they need. And so here on the right is a light micrograph of a cell that has been infected with rabies virus. And what we see here are dark staining inclusions in the infected cells. And these are called Negri bodies, after a pathologist who described them years ago. And he described them as being present in someone with rabies. And if you see these in a person who is suspected to have rabies, it is sort of diagnostic for having the disease. And they turn out to be where the virus particles are assembling. So that's an example of a factory. Pox viruses do this too. We talked about that. I showed you some pictures of cytosolic factories where pox viruses assemble. Polioviruses replicate their RNA on membranes. Many viruses do this. They synthesize their RNA on membranous vesicles that they actually induce in the cell. We'll talk about that on Monday. And these are obvious here. You can see the virus particles, all those small dots, but these double membraned vesicles, those are the sites of viral RNA synthesis. And again, we believe that one reason that RNA synthesis for many viruses occurs on these membranes is to concentrate all the components so they're not simply floating around in the cell looking randomly to interact with each other. You have the polymerase, the RNA, the NTPs, other proteins that may be needed. It doesn't make sense for them to have to just float around or be transported around and hit each other. So they're concentrated in very specific places so that, again, if it's dilute, it won't happen very quickly. A key aspect of assembly is that proteins have addresses to send them to the right place. Some people call these zip codes. And these include membrane targeting sequences like signal sequences, where if a protein is bound to be secreted or placed in a membrane, it has a signal sequence. It's translated in the rough endoplasmic reticulum, and the signal sequence guides the protein into the rough ER. And of course, those are at the end termini of proteins. They're removed after they're translated. There are also fatty acid modifications of proteins that make them hydrophobic and target them to membranes without a signal sequence. We'll see an example of that later. There are membrane retention signals where proteins have to stay in a membrane and not be secreted. And so those keep them in the membranes. Nuclear localization signals, which get proteins into the nucleus. So on the bottom are two examples of NLSs. A simple one, a 1, 2, 3, 4, 5, 6, 7 amino acid sequence. This is found in SV40T antigen because this is made in the cytosol. It has to get in the nucleus to stimulate DNA replication, and needs a nuclear localization signal because that 7 amino acids interacts with the machinery for import into the nucleus. And there are other sequences that are longer as well. This is from a cellular protein nucleoplasmid that happens to be longer. So many viral proteins need to get in the nucleus, but then they also have to get out as well. So we have nuclear export signals, similar idea that they have to interact with the export machinery, which now is inside the nucleus. And so they are recognized by one or more components from the export machinery. We'll see some examples of how these come into play during virus assembly today. So here's an example of localization to the nucleus. We have a cell, and it's just showing where various viruses have to get into the nucleus. We have our influenza virus. All the proteins, of course, are made in the cytoplasm. And this happens to be the nucleoprotein, which will coat the RNA. The RNA is made in the nucleus, so the nucleoprotein has to be imported. It has a nuclear localization signal that allows it to be bound at the nuclear pore and imported in the nucleus, where it can then be associated with RNA. The polyomaviruses like SV40, they are assembled in the nucleus. The DNA is replicated there, they're assembled in the nucleus. And so the capsid proteins are made in the cytosol. This is one of the capsid protein subunits. It's imported in the nucleus. Of course, it has an NLS. And same for adenovirus. Adenoviruses are assembled in the nucleus. And again, the capsid proteins have NLSs in them. Now, just because a virus has DNA replication in the nucleus doesn't mean it has to assemble there. We have actually learned recently of many viruses, particularly these giant viruses, that initially go in the nucleus, replicate their genome there, but then it's assembled in the cytoplasm. It's just another way to do things that works. You don't have to assemble in the nucleus. You can actually go out. It seems to us to be inefficient to do that. But as I said earlier in this course, don't use your judgment to think about what makes sense for a virus or not, because you're putting a human imprint on a virus, and it simply isn't human. And many viruses will assemble at the plasma membrane by a process called budding, which we'll talk about. And so there have to be addresses in the proteins to get them there. Here we have an example of a secretory protein made initially in the endoplasmic reticulum, and then it's transported up to the Golgi via vesicular transport. So a vesicle buds off the ER with the protein in it, then moves through the Golgi stack by a series of fusions, and then makes its way to the plasma membrane. And that vesicle will deliver the proteins to the plasma membrane. They move along microtubules to get from the Golgi to the plasma membrane. This little vesicle will fuse with the plasma membrane. Then you have the proteins displayed on the surface of the particle, and they can interact with nucleic acid components to get them packaged. And we'll see a more specific example of this later on today. Another key aspect of virus assembly is this concept of making sub-assemblies. You don't usually put the whole thing together at once. You make parts, kind of like an auto assembly line, where you make the door and then you attach it to the chassis and then you put wheels on and so forth. Similar thing happens with many viruses. This means a formation of discrete intermediate structures. And an example is shown here. It's a bacteriophage being shown, and I just think it's remarkable. The N bacteriophage is shown here at the bottom, and it's a tailed phage where you have an icosahedral head, a helical tail, and tail fibers, a base plate. These are all sub-assemblies that are put together separately. So here we have the head, and these numbers are simply the different proteins that go into making the assembly. So there's a whole series of reactions that occur to build this head, and then at the same time, you build the tail, you build the base plate first with a lot of different proteins, and then you build a protein shell around which the tail will be assembled, and that is stuck on the head. And then separately, the tail fibers are assembled and put on. So that's a very clear example of a sub-assembly, right? You make parts individually and then put them together. Now, why would you do that? Well, for the same reason that you make a car, I guess, on an assembly line, although I could be applying a human view to that, right? You should accuse me of that. This is how we view it, that it ensures orderly formation. You do it in an order, and that way things happen the way they should, and they don't get misassembled when you put all the parts together into a cell and hope that they come together in the right way. The other reason is for quality control. You make a tail, for example, and if it's broken, it doesn't get incorporated into the final virus particle. You can throw it away and use another tail assembly. And presumably there are mechanisms that will distinguish when a part is not made properly. So those are sub-assemblies, and they happen in the replication of many viruses that we're going to look at today. How do you make sub-assemblies? Here are three ways that you can do that. First, you can make individual protein molecules. So an example is the capsid subunit for SV40, which is a pentamer made of 5 copies of VP1, and these are used to build the complete virus particle. And this is made, as I said, of 5 copies of VP1 and a minor protein, VP2/3, which is in the middle there. And these are simply translated individually. You translate VP1 in the cytosol, you translate VP2 and 3, and they will assemble. Remember, the sequence of these proteins dictate their ability to assemble. That's part of the beauty of this icosahedral symmetry. It lies in the amino acid sequences of the protein. You can also make polyprotein precursors. Some viral genomes, like RNA viruses, have a problem. They have to make many proteins from a single RNA. And one of the solutions is to make a polyprotein, which is a single open reading frame. In eukaryotic cells, you can't translate more than one protein from a single mRNA. So if you have a one viral RNA, which is the genome, it's hard to make more than one protein from it. So one strategy is to make subgenomic mRNAs. The other is to make a polyprotein, which gets chopped up by proteases. So that's here an assembly from a polyprotein precursor. We have, as an example, poliovirus, where ribosomes are translating the capsid protein gene, VP1, 2, and 3. And this folds in a way that it can be cleaved by the viral protease and forms the structural unit. So that's a sub-assembly. It's going to go on to form larger and larger assemblies to make the virus particle, but it's made it from a polyprotein precursor. Now, the third mechanism here is chaperone-assisted assembly. Chaperones are cellular proteins that help reactions go properly. And in this case, they help the proteins fold in the right way, even though I said that all the information for protein folding is in the primary sequence of the protein. Sometimes it needs some help with the folding from a chaperone. For example, if the protein is particularly large and the interactions are complex, the chaperone may help. These chaperones can be viral or they can be cellular, as I mentioned earlier. And here we have an example of the adenovirus structural protein, which will eventually go on to form the hexon trimer. And this will be a subunit of the virus capsid. Its assembly into a trimer is helped by a viral chaperone called L4. But there are lots of other chaperones as well that assist assembly. And I don't expect that you know what these chaperones are, just that you understand that they are there to assist in assembly reactions. They can be viral or they can be cellular. And we have 4 different examples here. A phage assembly of its capsid, assembled by chaperones, GROW-EL, for example. It's a bacterial protein. We have next poliovirus assembly, which I just showed you, being helped by a number of chaperones, including HSC70 and P23. And again, they're helping it to fold in a way that will allow cleavage. Here we have retrovirus assembly being helped by a chaperone tri-C. And this starts with a single polyprotein, the GAG polyprotein, and is folded up into a capsid, as we'll see later. And at the bottom, even the SV40 assembly, the polyomavirus assembly from that pentamer of VP1, is assisted by HSC70. And look what's crept in here. Large T antigen. It even has a function as a chaperone for capsid assembly, among all its other functions being involved in DNA replication and transcriptional regulation and so forth. That is sequential stages of assembly. So let's look at an example of this, poliovirus here, which has been really well studied, so we understand all the steps involved. And this is the entire replication cycle. So all happens in the cytosol. Viruses bind receptors. The receptor itself catalyzes conformational transitions that allow the RNA to come out of the particle. This is the receptors makes this virus, it makes it unstable. And so the RNA is coming out through a pore that's formed in the capsid by the receptor, and it's now in the cytoplasm. It's translated into a long polyprotein, which is then cleaved by virus proteases, and then the P1 region, the very end terminus, goes on to form the capsid. And that folds into the 5S structural unit, VP1, 2, 3, and 4, and that folding, as I showed you previously, is assisted by a chaperone. The 5S structural units, so that's one copy of each protein. 5 of those then associate into what are called pentamers. 5 copies pentamer. So that's sequential assembly. One structural unit, 5, and then we have formation of an empty capsid, which has 12 of these pentamers, 12 times 5, 60 subunits. That empty capsid doesn't have RNA in it, so it's not going to be of much use. And so we don't think it's actually an intermediate in the assembly, but rather the RNA joins with these pentamers and forms the capsid, which then can mature by cleavage of one of the capsid proteins and will exit the cell. So it's a relatively simple series of assembly steps. 5S structural unit, pentamers, and then the capsid with the RNA. Here's another one, a more complicated virus that illustrates a couple of other concepts that are interesting. And this is herpesviruses. And these are, again, viruses with DNA genomes. They replicate in the nucleus. And in fact, they assemble new virus particles in the nucleus. And so DNA is replicating in the nucleus. We're not showing it here, so you can see genomic DNA. But all the capsid components are made in the cytosol, and they have to be imported. And some of them are shown here. VP5 hexamers are a major part of the capsid. And look, there's one portal that is made. Remember, each capsid has one portal, and it's shown there. So they're all imported into the nucleus, so all of them have nuclear localization signals. Now, the way this virus is built is quite interesting. First, what is built is a procapsid, and that's the shell, but inside of it is a scaffold. Because the idea here is that this is so large that it can't assemble on its own. It needs help. So an internal scaffold is built rather than the scaffold on the external part of buildings that are used to build the building. This is built on the inside, and it's composed of all these squiggly proteins shown in black, and they're VP22, 23, 19, et cetera. All of these form the scaffold. And so then the scaffold is built and the shell forms around it. And all the interactions of the subunits of the shell then make it very stable, so then you can get rid of the scaffold. So how do you get rid of it? It's inside a nice, stable capsid. Well, the scaffold itself has a protease built into it, such that when the capsid is complete, the protease is activated, and it digests away the scaffold, as you can see here in the next step. The protease is VP24, and the peptides come out the portal, and then the genomic DNA is imported into the capsid through the portal. All right, so this procapsid is an intermediate structure. It's a sequential assembly because we're doing things in step. The procapsid is intermediate. We get rid of the interior scaffold, put the DNA in it, and then we have a virus particle that's going to be infectious. Now, this is only the nucleocapsid. It has to acquire a membrane. We'll look at that later. We'll also look at how the DNA gets specifically into the capsid. All right, so that's sequential assembly. For many viruses, we have an aspect called concerted assembly, where particle forms more or less all at once, although there are sequential steps for the formation of the components still. So it's never purely concerted. But this is meant to emphasize that when virus particles form by budding, you're basically making the particle all at once. So this is influenza virus, where the RNA replicates in the nucleus. So the incoming genome, which is not shown here, goes in the nucleus. The genome is transcribed to make mRNAs. They're exported. They give rise to a variety of proteins, including the glycoproteins that are produced in the ER and shipped up via the Golgi to the plasma membrane. So here they're inserted into the plasma membrane. At the same time, the viral RNAs are replicated, full-length copies are made, negative strands. They have to be coated with the nucleocapsid protein, which has to be, of course, imported. It coats the RNA. And then we have an M1 protein, this blue rectangle, which is, again, made in the cytosol. It goes in, it binds to the RNA, and that protein, the M1 binds a second viral protein called the nuclear export protein. And as you might guess, that gets it out of the nucleus into the cytosol. The M1 protein will then associate with the budding site by a mechanism we'll see in a bit, and then that whole assembly buds out to form a virus particle. So that's why it's concerted assembly, because this budding forms the virus particle. But again, there are sequential steps before that, because you have to make the RNA, you have to make the RNA M1 complex, you have to make the glycoproteins in the membrane as well. So many viruses do that. A little more detail on how the glycoproteins are incorporated into the particle. The influenza viruses, remember, have a membrane in which they're embedded a number of viral glycoproteins. We've talked a bit about the hemagglutinin in terms of it binding to sialic acid, which is the cell receptor. There's also a neuraminidase, another protein which happens to be an enzyme, and we'll talk about that later in terms of pathogenesis. And then there is an ion channel, which we talked about in terms of virus entry. It lets protons into the interior to help uncoat it, the particle. So here's a schematic of the HA protein. And remember, the HA protein is on the particle as a trimer. It's shown in the upper right in its three-dimensional structure. And the globular head is what binds the receptor. And at the bottom is just a line drawing of this protein to show you how it works. It's a transmembrane protein. It's initially put into the plasma membrane and has a transmembrane sequence. And the red is the extra virus part of the HA. Because it has a signal sequence, that little squiggly purple part, and that's what initially gets it into the ER. It's cleaved off, so it's not present in virus particles. Signal sequences are removed by host enzymes called signal peptidases. This protein is highly disulfide linked, as you can see by the yellow disulfide bonds. And there is the fusion peptide, very near the membrane. And in the immature HA, it is not available because it's not cleaved. And that orange arrowhead is the cleavage site for proteases that will cleave there and make the N-terminal fusion protein available. And remember, that's hidden until low pH is encountered in the endosome, and then it's exposed and inserted into the membrane. But here is a nice illustration of how cleavage is required to expose the fusion peptide. And for most influenza viruses, as we'll see later, that cleavage occurs extracellularly. And the protease that does it appears to be restricted to the respiratory tract, which is why the virus only replicates there most likely. And the influenza HA, as I mentioned, is destined for the plasma membrane, but it's initially made in the endoplasmic reticulum. The ribosome binds to the ER, and the signal peptide directs the translocation of the protein into the lumen of the ER. As it's made, the HA oligomerizes right away, it forms trimers, and then it's shipped up towards the plasma membrane through the Golgi network, which involves a series of vesicles budding off the ER, then fusing with the cis, and then moving to the different stacks of the Golgi, and eventually to the plasma membrane. But as proteins move through this secretory network, they undergo a variety of modifications that are important. For example, oligomerization, which is important. It has to be a trimer. There is a lot of sugars added. There are a lot of sugars added and taken off at different parts of this pathway. Sialic acid is one of them, until we get to the mature protein where we have a lot of sugars on it. In this case, it's already cleaved. Some influenza viruses are actually cleaved as they're transported through this network and not depend on cleavage when they're outside of the cell. This first question is, sub-assemblies are involved in which of the following types of virus particle production? Concerted assembly, sequential assembly, assembly lines, chaperone-assisted assembly, or all of the above? So it's all of the above, which most of you got. So sub-assembly is simply less than the final virus particle. So for poliovirus, it's, for example, a 5S or a 14S subunit. And so they can be involved in any of these. Influenza viruses make sub-assemblies. Sequential assemblies, where you put things in step, they involve sub-assemblies or assembly lines. And chaperone-assisted assembly can involve sub-assemblies as well. So they're all right. Those are how you make the protein shells. How do you put the nucleic acid in? That's what we call genome packaging. And so the problem is to distinguish viral genomes from cellular, either DNA or RNA, depending on what kind of virus it is. And the solution seems to be that there are sequences in the viral genome, whether it be RNA or DNA genome, and we call these packaging signals. Right? It's a little bit of a jargon there, packaging signals, because to the person who's never thought about viruses, packaging doesn't make much sense, right? But we're talking about packaging the nucleic acid into the particle. So here, at the top is shown a budding retrovirus, and the genome in green is being packaged into the particle. How does it ensure that only the genome is in there and nothing else? And the solution seems to be specific sequences that are present only in the viral nucleic acid and not in cellular nucleic acids. And these are important because if you want to do gene therapy, let's say you want to put a gene in a virus to replace a gene that someone is lacking. You have to put a packaging sequence in the gene. And so people studied packaging sequences for 40 years without that goal in mind. They just wanted to understand how nucleic acids got specifically into the particle. And then parallel, people were learning that you could use viruses as delivery systems for genes. And then the two came together. The people who studied the packaging said, you have to put these sequences in your vectors. And we'll talk about that as the last lecture in this course. So here are some packaging signals in DNA genomes. The adenovirus is an SV40. You see adenovirus at the top. We're at the very left end of the genome here. This is the left end, and that's one of the two inverted terminal repeats that are present at either end. And that's one of the two origins of replication. There's one at each end. Packaging sequences here between 100 and 500 base pairs from the left end. So it's very close to the end. And it's a series of, they're pretty complicated. It's a series of overlapping sequences. All these blue arrows are the packaging sequences. They overlap with these enhancers. Remember, enhancers are long-distance stimulators of transcription. Here's the first transcript that's made, the E1A transcript, and these are enhancers of that. So they're overlapping with that. And in this case, for adenovirus, these sequences are recognized by a specific structural protein, 4A2, that is bound to the viral DNA. So 4A2 binds these sequences, and then 4A2 is recognized to be pulled into the capsid. And at the bottom is SV40 packaging sequence. Again, here's the origin of replication. Remember, SV40 is a circular DNA. So the origin is, if we put the origin at the top, then there it is, base 1, and the packaging sequences is just to the left of it. And again, it's near an enhancer, which is an enhancer for the early transcriptional unit. So in general, these sequences are, again, unique to the virus. They're not found in cellular sequences, and they're bound by viral proteins, and that's how the nucleic acids are pulled into the particle. The herpesvirus packaging strategy is really wonderful. It's shown here. And remember that herpesviruses replicate by rolling circle. The DNA circularizes when it comes in the nucleus, and new genomes peel off as it replicates. The polymerase replicates around the circle. And you get concatamers, which means multiple big DNA, long DNA molecules with many genome equivalents in them, all covalently linked. You can see 2 genome length DNAs right there at the top left. So how do these get into the particle? Well, at the left end of the viral DNA, it's shown here in panel A, there are a couple of packaging sequences called PAC1 and PAC2. So that's the whole herpes simplex genome there in panel A. And then at the very left end, you have packaging sequences. And these packaging sequences interact with the portal. Remember, there's one unique portal in the particle. This is now assembled in the nucleus. It's been assembled, the internal scaffold is removed, and there's that one portal, and the packaging sequences interact with a variety of proteins. So in this expansion in the top of panel B, step 1, there's the portal, and there's the viral DNA with the packaging sequences, PAC1 and PAC2, and they interact with a series of viral proteins which also interact with the portal. So if you have multiple genome lengths at the one end of the genome, of each genome, you're going to have this packaging sequence repeated. It's going to be present in all the genomes, of course. So once this packaging sequence is bound by the portal actually has a motor in it that pulls the DNA inside of the capsid. It's actively pulled in by an ATP-dependent motor, and this is very common for DNA-containing viruses, particularly bacteriophages. They have these motors, which have been extensively studied, that can package DNA by pulling it in the head, and they do it at high pressure, incredibly, like 900 pounds per square inch, right? It would blow your tires up if you blew them up. Your tires are 30 pounds per square inch. So think of this. 900 pounds per square inch inside of these capsids. And the capsid is so stable it doesn't explode. And then when the portal docks onto the nuclear pore, out comes the DNA because it's under such high pressure. It's brilliant. So this portal motor brings in the DNA. And how does it know how much to put in? Well, there are two controls. One is when it reaches the head, when the head fills up, when the capsid fills up with DNA. We call that the head full hypothesis. But the other signal is that when the next packaging sequence arrives at the portal, then the portal knows that we've got enough DNA in there. The DNA is cleaved by an endonuclease, and now we have a head full of DNA in the capsid with the packaging sequence there. And then the other DNA that's left, the rest of the concatemer, it has its own packaging sequence, and that can be put into another virus capsid. So recognition of this packaging signal by the portal, pumping in the DNA and cleaving when you have a head full of DNA in it. So those are two examples of DNA packaging signals. How about RNA viruses? They have packaging sequences as well. The best characterized are in the retroviruses. That's good because many of those are used for gene therapy applications. Here is the packaging signal for HIV-1. So this should, on panel A, this should look familiar. We looked at this last time. This is the very 5' end of the viral genome, the mRNA. So there's base 1. There is the primer binding sequence, the PBS, where the tRNA binds, where you will get the first reverse transcription. And the packaging sequence psi is shown as psi, is shown right there, that red psi. And that's it. That's the packaging sequence in those bases. You can see it's highly structured. This is necessary for packaging, but it's not sufficient. You need some other signals in the genome as well. So if you're building a vector, you need this and something else. And the way this works is twofold. First of all, this contains the stem loop 1, which makes this kissing loop complex we talked about last time. Remember, the retroviruses have 2 RNAs in their particles, and the 2 RNAs interact via these loops on stem loop 1. They actually base pair. That's what keeps them together. It's then recognized by a viral nucleocapsid protein, NC. So on the right side here is the GAG protein, the very first protein in the retroviral genome when we look at the simple genomes. It's composed of matrix, capsid, and nucleocapsid, and we'll revisit these other 2 proteins. But the nucleocapsid at the C-terminus has 2 sequences that bind RNA, so they will recognize this 2 RNAs, and they will bind the RNA. And so what's formed is shown at the bottom here. So we have a nucleocapsid, which is this circle, white circle. The nucleocapsid is binding the RNA, and that's attached to a capsid protein, the purple one, and then a matrix protein. And this whole thing, as you'll see in a moment, is going to form the viral capsid. But that's how the RNA gets into the capsid. Influenza virus illustrates another issue, and that is, if you have a virus with a segmented genome, how do you make sure that all of the right number of segments get into the particle? So for a single genome, it's kind of easy. You just have a packaging signal that pulls it in. But here we have 8 segments in influenza viruses. And how do they get in? Well, so for many years, people thought it was just random. The virus just packaged any 8 viral RNAs in the infected cell. So imagine in the cytosol, you have all these RNAs that have been coming out of the nucleus. And when you bud, you just pick 8 of them. And maybe many particles will not be infectious because they're missing the right segments. And in fact, when you do the math, if you have 8 segments and you just incorporate them at random, 1 in 400 are going to be infectious. So 1 in 400 times you'll get the right mix. And that is near the particle to PFU ratio of the virus. Remember, particle to PFU, how many infectious viruses per particles? So that could be a random thing, but people weren't happy with that, so they kept looking. And now there's evidence for a specific packaging sequence on each RNA segment. So let me show you how that works. So here on the right are the 8 influenza virus RNAs. They each encode 1 or a couple of proteins. And at their ends, there seem to be specific packaging sequences, which are shown in blue here. And the numbers just show you how long they are. It doesn't matter the length. But each segment appears to have a specific sequence that directs it to be incorporated into the virus particle. And so you always get 8 segments, and apparently these segments, these sequences are interacting with each other and maybe with proteins to make sure that only one of each of the 8 segments get into the particle. Part of the evidence for that is you can swap some of these signals. So you could take these signals from PB2 and put them onto PB1, and you still get the proper packaging. The other interesting aspect of influenza virus packaging is when the viruses bud from the cell surface, and that's shown at the upper left here, you can see the formation of a particle as it's budding off. The RNAs are always parallel and perpendicular to the membrane, just as they're shown here. So the 8 segments, parallel and perpendicular to the membrane. So they seem to be lined up in a way which may be mediated by these sequences. And if you take an electron micrograph, you can see in each particle that the RNAs are all parallel because they're all little circles there. Think of it as a cross section. They're all parallel. So you don't see any of the RNAs on their sides. So there is a specific packaging sequence that is at the ends of each RNA that mediates only one of each RNA getting into the particle. So that's one way to do it if you have a segmented genome. The other is to have selective packaging, which is illustrated by this bacteriophage called Phe6, which is a bacteriophage. It's a spherical bacteriophage. They don't all have the heads and the necks and the tails and the tail fibers. And this has a double-stranded RNA genome. 3 segments of double-stranded RNA, the small, the medium, and the large segment. And just like the reoviruses, it has 2 shells. It has an outer and an inner capsid. And they're the 3 segments of RNA. So the way this packages 1 copy of each double-stranded RNA, you have a serial dependence of packaging. So first, the small segment goes in. Must be some specific signal that gets it in, and the other 2 can't get in unless the small segment is there. So next, the medium segment goes in, and finally the large. So there's a sequential packaging, and this virus, interestingly, has a particle-to-PFU ratio of 1. So maybe this is a good way to package because you always get the right number of particles in. There aren't many broken particles here with this virus. So here's a question about packaging. Bless you. Packaging signals on viral something interact with viral something else during virus assembly. And we can have the 2 words being lipids, proteins, sub-assemblies, genomes and proteins, proteases and membranes, and proteins and genomes. So the answer, of course, is genomes and proteins. Packaging signals on viral genomes interact with viral proteins during virus assembly. And some of you picked proteins and sub-assemblies, but there's no genome in there, so we have to have genome. Proteases and membranes, again, there's no genome. Proteins and genomes, a packaging signal is not on a protein, it's on the genome, so that's reversed. Listening to someone sneeze before, we're in the middle of flu season, so you might have influenza, but you would know it. You wouldn't be here if you had influenza, you'd be pretty sick. But it turns out that we'll see later, it's not how the virus is spread. Sneezing isn't the major way of transmitting influenza. It's coughing and just speaking. It turns out when you speak, you emit huge aerosols with lots of particles, especially the P's, right? P, and that shoots out a lot of virus. So CDC tells you when you sneeze, you're supposed to put your... Apparently you don't shoot out a lot of virus by sneezing. We'll talk about it later. But the sneeze reminded me of that. All right, getting an envelope. Almost everything reminds me of viruses. That's just the way it is. So, how do you get an envelope? There are 2 ways, 2 general ways. First, you make the internal structures, like we did with influenza virus. We made the R and P, and then it buds off the cell. But there's another way we see with other viruses. We say simultaneous with assembly of the internal structures. What does that mean? The whole structure forms as it buds. You don't make the internal structure first. It only forms upon budding. All right, so here are 4 ways that viral proteins drive budding. As we'll see in a moment, budding requires cellular proteins as well. You would expect that. But a major component is no bud is going to happen without viral proteins. And so, for example, in some viruses, the glycoproteins and the capsid are needed for budding. So you have to have glycoproteins here in red on part 1, but you also need a capsid, and then it will bud out. For other viruses, the matrix protein is enough to drive budding. This is the case with some retroviruses. Sometimes envelope proteins alone will drive the formation of a bud. And this is actually the basis of some vaccines. We make some influenza vaccines by simply producing the HA protein in cells. It forms particles, virus-like particles. They bud off the cell, and they're just HA with nothing inside, so they're not infectious. So there, that's the glycoprotein will drive budding. And sometimes you need multiple components. Matrix alone is not good enough. You need glycoproteins or a nucleocapsid. Various viral components will drive the formation of a bud. And so you can produce, for example, in retrovirus, in the case of retroviruses, part 2, you can just produce a single protein, and the GAG protein alone will make virus particles. Matrix is a sub-fraction of the GAG protein. And so let's look at budding in a couple of viruses. Here is influenza virus budding. And this is an example of the first mechanism of budding, where the internal structure, assembly, and budding are separated, both spatially and temporally. So here we have, as we saw before, the assembly of nucleocapsids in the nucleus. They are exported by the virtue of the nuclear export protein that gets this RNA M1 complex out. That then makes its way to the plasma membrane. Now, I showed in a previous slide basically the same thing, but there was none of this microtubule. And this is important because the RNP M1 doesn't just diffuse from the nucleus to the plasma membrane, it moves on microtubules, assisted transport, and that requires energy. And eventually they reach the plasma membrane and interact with the glycoproteins that are already there, and that forms the bud. So in other words, the internal structure forms first. We're only showing one RNA here, right? But there are 8 of them. They form first, and then the particle buds. So you're doing this in 2 separate steps. And that's going to be in contrast to viruses where it all happens at once. Now, an important part of this budding is getting the components to the right place. Now, for example, we know that the glycoproteins are targeted to the plasma membrane because they have signal sequences that send them from the ER through the Golgi to the plasma membrane. But what about the M1 protein? How does it know where to go? So here are 2 examples of how it knows where to go for flu, influenza virus, and vesicular stomatitis virus. These are 2 proteins that have to make their way to the membrane. And the influenza virus M1, we just saw how it comes out of the nucleus bound to RNA and goes to the plasma membrane where the viral glycoproteins are. So the M1 protein is shown as a line here, and you can see various regions indicated. Nuclear export sequence, so this is part of the mechanism for this protein getting out of the nucleus. Has a nuclear localization signal because, again, it's made in the cytosol, it has to get in the nucleus. And then it has sequences that will bind to the RNA, the ribonucleoprotein. So M1 has sequences that bind it to the RNA. And finally, it has hydrophobic regions. Those are in orange. These things are all overlapping, right? They're not just discrete sequences. Orange hydrophobic regions says lipid binding. That's what gets the M1 to the membrane by virtue of these hydrophobic sequences in the protein, which will have affinity for the membrane, and it will go up to the membrane. If there are viral glycoproteins there, it will become part of a new particle. If there are not, the M protein will move along the membrane until it encounters these glycoproteins, probably via their cytosolic sequences that are below the plasma membrane. So that's influenza virus. The M protein of VSV has a similar property in replication. It is targeting the plasma membrane where viral glycoproteins are inserted. And you can see it has a hydrophobic region again, which is for membrane binding. And it has another region for binding the RNA. So this protein binds the RNA and carries it up to the membrane. All right, so that's the formation of an envelope particle in 2 different steps. Now here's the example where it all happens at once, and that's the retrovirus. So now remember, the retrovirus starts with the provirus. You have integrated into the cellular DNA, the viral genome, the DNA copy. It is transcribed into various mRNAs, including a full-length mRNA that's going to get packaged, as well as other mRNAs that give rise to the various proteins that are needed. So here in the nucleus, we make the mRNA for the genome. It's come out of the nucleus, and we're showing it here. It's a dimer, and it is bound to the nucleocapsid protein. And remember, I showed you before how the nucleocapsid protein binds to the RNA, and the nucleocapsid is part of the GAG protein. It's shown at the right here. The GAG is made as a precursor, and it is then assembled onto the RNA as a precursor. So these little proteins that are on the RNA genome below step 1, nucleocapsid, that's the white one. The color coordinated with this line diagram at right. Then the capsid protein is the brown one, I guess. And then finally the matrix is the blue one. So by virtue of NC binding RNA, it brings capsid and matrix along with it. This matrix protein targets the plasma membrane by a mechanism we'll look at in a moment. So these RNA protein complexes are moving to the plasma membrane. At the same time, the glycoprotein has been inserted there because the virus has a glycoprotein in the envelope, TMSU. And these sub-assemblies, matrix, capsid, NC, all line up at the plasma membrane. Look at that. So we have the blue is the matrix protein, which is going to eventually form a shell beneath the membrane. See that? And below it is the capsid. The capsid is going to form the isometric capsid of the particle. And then we have the nucleocapsid. You know, every nucleocapsid protein, NC, is not bound to an RNA. We only need 2 RNAs in this particle, so most of them are not. So you can see they're interspersed with matrix, capsid, NC subunits where there's no RNA bound. But these initially coalesce at the plasma membrane, then the virus particle starts to bud out. But there's no virus yet. See, for flu, you would make a virus particle right away. Now you even have the formation of what looks like a particle, but this is not infectious, step 3. This is not done yet because the proteins haven't been arranged properly. We have all of the matrix protein is there, the capsid is beneath it, but it's just a spherical shell. We have reverse transcriptase and integrase and protease. Those are brought in when the GAG translation keeps going. GAG protein ends with a stop codon, and at a small percentage of the time that stop codon is suppressed and you get a GAG-POL fusion. And so that brings the enzymes into the particle. So the matrix goes up to the membrane, brings in capsid, NC, then protease, reverse transcriptase, and integrase. You don't need a lot of that, so that doesn't happen very often. But you can see they're incorporated into the growing particle, and at some point the particle buds off, but it's not yet done yet. So here is where the particle is formed at budding, but you have to go an additional step. The protease becomes active now, that's the yellow sphere, and it cleaves the bonds between matrix and capsid and NC, and in some cases protease RT and integrase and so forth. And that matures the virus particle. You can see it's drawn now as having a isometric nucleocapsid, and that's infectious. All right, so that's what I mean by the internal assembly and budding are spatially and temporally coincident. You don't actually form the particle until it's totally budded off and the protease has become active. But you can see here a lot of interesting points, in particular how the RNA is brought in and how these arrayed proteins form the growing particle. I should point out here, GAG alone, if you just make this protein by itself, you will get particles. You will not have envelope in it unless you include envelope. And you could do that, and then you could make particles that are empty with envelope. But if you want, then you could add a gene to this. And if you have a packaging signal on your gene, you could then get it incorporated into virus particles because the packaging signal will be recognized by nucleocapsid protein and brought in. We'll talk about that later, as I said. All right, so matrix, how does it get to the plasma membrane? I promised to show you that. That's shown here. Here's the GAG protein again, matrix, capsid, NC. The NC, I told you before, has RNA binding sequences in it. So this part of the polyprotein binds the RNA. The matrix protein at the N-terminus is myristylated. A myristate is a lipid that can be covalently attached to amino acids, typically at the N-terminus of proteins, typically an N-terminal glycine. That will interact with membranes. It's highly hydrophobic. It's a lipid. And there are also other membrane binding sequences within this. And if you change this glycine so it can't be myristoylated, GAG can no longer interact with the plasma membrane, the inside part of the plasma membrane, and virus particles don't form. So that's very important. This is what myristate looks like. So again, it's a lipid linked to a protein covalently. So and this is one of several lipids that can be attached to proteins to target them to membrane without a signal sequence. Signal sequence is what drives proteins to the ER, right, that are destined for the plasma membrane. But here's another way to get proteins there without a signal sequence, that is by putting lipid on them. And so you could make a protein in the cytosol and modify it with lipid and target it to the plasma membrane. If you didn't have the lipid, it would never get there, because the only other way to get to the plasma membrane is by a signal sequence, and that can only be added when the protein is put through the ER. All right, the last aspect of budding is discovered maybe 15 years ago, and it was discovered as virologists were making changes in the GAG protein of HIV and a murine retrovirus, MLV. And they found that amino acid changes in this part of GAG, P6 or P12, depending on the virus. It's this small protein. You know, here we have matrix capsid NC, matrix capsid MC, but there are these little interspersed proteins. And people used to say, they're just linkers. They're there for flexibility. But it turned out if you made amino acid changes in them, you got these budding viruses that never detached from the host cell. They were held on by these stalks or membrane tethers. See them there? Again, amino acid changes in these regions, P6 or P12 of GAG, cause this. And it turned out that, well, so these were called late domains because they affected a late stage of virus assembly, the budding stage. They were ultimately found in many other viruses, regardless of whether they're plus or minus stranded. As long as they had an envelope that you could find a protein which, when altered, would give you this kind of phenotype. And finally, people discovered that these proteins, P6 or P12, interact with cell proteins that are involved in vesicle trafficking, and they're absolutely required for virus release. So what are these cell proteins? They are the ESCRT pathway. ESCRT, endosomal sorting complexes required for transport. This is a cellular system which is needed for any kind of function where membranes have to divide. For example, when cells divide during mitosis, you got to split the two membranes off. That's mediated by the ESCRT proteins. Shown here is a little coil, ESCRT3, and that's called abscission. The formation of exosomes or multivesicular bodies involves membrane fusion. That also involves the ESCRT pathway. And there are many different proteins that constitute this pathway, ESCRT1, 3, ALEX, VP4, etc. And what they do is they mediate these processes. So the retroviral proteins, P6 and P12, interact with components of this ESCRT pathway, and they divert it to the cell surface to form a bud and to pinch off that bud when needed. You can see ESCRT3 here is eventually going to pinch off that virus so it can be released. And if you alter P6, which is interacting with, can't see it here, but P6 is interacting with the ESCRT machinery to bring it here. If you alter P6 so it no longer interacts with ESCRT, then you can't do the final step of the bud, which is the pinching off and the release. You end up stuck at this middle stage, which is the virus on a stem. So just by making little changes in amino acids in a virus, and people weren't looking, to find this. They said, what do these little proteins do? So never assume that a little protein is just a spacer or a filler. You never know. This started a whole new area of research and revealed this pathway, which is really important for many cellular proteins. And it seems most budding viruses are dependent on this pathway for budding. Now, the last aspect of budding is that it doesn't have to happen at the plasma membrane. It can happen at virtually any other membrane in the cell, including the nuclear membrane. Herpes simplex viruses bud from the nuclear membrane. It can be budding from the endoplasmic reticulum. It can bud from the Golgi, or it can bud from the plasma membrane. Right? So any of these membranes will suffice. And it's interesting to think about how a virus would get out of the cell if it buds from one of these internal membranes. And let me show you an example of that. Well, we'll have to have a quiz first, and then we will get to that. All right, next question. What statement about viral budding is incorrect? The envelope can be acquired before or simultaneous with assembly of internal components. The spike glycoprotein can drive budding. No host proteins are involved in the budding process. Lipids assist structural proteins to interact with the membrane, and the viral membrane can be acquired from the nucleus, Golgi, or plasma membrane. The answer is C. No host proteins are involved. That's wrong. It's incorrect. Absolutely. ESCRT proteins I just told you about. Everything else is right. You can get the envelope before or after assembly of internal components. So before assembly of internal components, that's the retrovirus example. The glycoprotein can drive budding in some cases, and the membrane can be acquired from any of those locations. It's absolutely right. All right, so let's look at a case where a virus buds from the nucleus. This is herpes simplex virus. And see what it has to do to get out without losing its membrane. So here we have the capsid assembled in the nucleus, which we talked about earlier, right? It's assembled with a scaffold, the DNA gets put in through the portal, and then it buds out of the nuclear membrane. So that's the nucleus on the left there. It's the nuclear membrane. ER is right next to it. So that budding gives it a membrane, puts it in the ER. And by the way, these are all electron micrographs of each step. So you can see it actually happens. But how do you get out of the ER? Well, the only way out is to fuse, and then the virus loses that membrane that it acquired. So here's the nuclear capsid again, with all those proteins around it, by the way, which include VP16, which is an important transcriptional activator. So then that goes into the Golgi. It buds into the trans-Golgi. And now you have an envelope virus in the Golgi. All right, so if you just fused out again, you would lose the capsid again, which would work, I think, because then you could bud out at the plasma membrane, but that's not how it works. This envelope virus buds out of the Golgi in a vesicle. So now we have two membranes on it. That vesicle then goes to the plasma membrane and fuses and releases the herpes simplex virus with its own envelope. And if you trace back, where did that envelope come from? Where did this come from? Can anyone tell me? What's the origin of this envelope as far as the cell goes? It's the Golgi, right. See, it's right there. It was maintained. So these viruses that do budding from different places have to go through unusual machinations. The other thing that happens as viruses move through the secretory pathway, they can undergo other structural alterations needed for infectivity. And this is an example with dengue virus, a flavivirus. Remember, these viruses have a glycoprotein that's sitting flat on the surface. It attaches to the receptor. On low pH, it moves up, and the fusion protein gets inserted into the membrane. Now, as this virus is assembled, it gets assembled in the ER, acquires its membrane there, and begins to move through the trans-Golgi network. The pH drops, and there's a cleavage event that occurs. Now, the virus particle, as initially made, is immature. It's shown in the lower right here. The glycoproteins are sticking up, and they can't be that way, so that has to be fixed. Plus, there's a fusion peptide up there. It's covered by this blue protein, PRM, and the blue protein masks the fusion protein, so this virus doesn't just keep fusing throughout its movement in these membranes, right? As the pH drops, these proteins flatten on the surface that expose a cleavage site for a furin protease on PRM, And then when these particles are released, this cleavage occurs here in the trans-Golgi, see furin cleavage. And then when the particles are released, PR comes off, and now the glycoprotein is spring-loaded. It's flat on the membrane, ready to pop up when the pH drops. The fusion protein is hidden, and that's all mediated by movement through the trans-Golgi. And this happens for a lot of viruses. They're spring-loaded by these kinds of movements. All right, the last few minutes I want to spend talking about how viruses get out of cells. One way is very cool, exemplified by vaccinia virus. They're actually propelled on actin tails. So here's a vaccinia virus which has matured in the cell. It's got two membranes. It moves to the plasma membrane on microtubules by motors. One of the outer membrane fuses with the plasma membrane. Now you have this extracellular virus here. It sits on the cell surface, interacts with a receptor that stimulates a signaling pathway through SRC, which triggers actin polymerization. And that's what these lines are. And these actin polymers make the virus get pushed away from the cell. And that enhances the ability to infect another cell. And you can see those tails on these micrographs here. Here's a fluorescent labeled tail with the virus particles at the ends. And here's one showing you, here's the virus particle at the top left in B, and that's the actin filament in the cell itself. It's remarkable that this gets done. So apparently this virus requires to be pushed from the cell to go to a new one. Many viruses are simply lyse the cells that they infect. Here's the picture I showed you earlier of poliovirus. It induces apoptosis or necroptosis. They simply break cells and the membranes rupture and the virus particles release. Many viruses encode proteins that cause membranes to rupture, like viroporins. And often many viruses shut off protein synthesis, and that makes membranes lose their integrity. So many viruses simply lyse the cell. We call them lytic viruses. And remember that because we're going to talk about that in terms of viruses causing disease later on. But we now recognize that there are many examples where viruses are released from cells without lysing them. And they're released in membrane vesicles. And there are two examples here. And these are viruses that are non-enveloped, like polioviruses and related viruses. They can be released in these double membrane particles, which are made by autophagy. When a cell is stressed, it makes these vesicles and encases contents of the cytosol and shuttles them out of the cell, sort of to recycle them as the cell is dying. And many viruses get trapped in them and are released from within these vesicles. And now, more recently, we understand that many virus particles are released from cells via multivesicular bodies. They bud into these vesicles, acquire a membrane, and then are released as exosomes. Exosomes are little vesicles released from cells normally that have components in them needed for communication and so forth. They can enclose viruses as well. Even polio and related viruses are shown to do this. This is really interesting because these membranes don't have any glycoproteins in them. So people are really interested in understanding how they attach to cells and infect cells without glycoproteins. All right, next time we will end our discussion of the reproduction cycle by talking about the kinds of changes, metabolic and otherwise, that are induced in infected cell by infection.